All right. Hi, my name is Pilar Magoulas. I'm a genetic counselor at Texas Children's Hospital and Baylor College of Medicine, and I'm excited to speak to you today on understanding your genetic test result. And so what I'd like to do today for this webinar is to first review the basics of genetics, describe genetic variants, go into the genetics of HADS, different genetic testing options, reviewing what you might see on your genetic test result, and then some frequently asked questions that we typically see in our genetics clinic. So to start off, I want to first start with chromosomes. And so chromosomes are these structures that are found in every cell of our body. They are numbered according to size. So 1 is the largest, with 21 and 22 being the smallest chromosomes. You can see that we have 2 copies of every chromosome because we get 1 copy from mom and 1 copy from dad. The 23rd pair are the sex chromosomes, so females have 2 X chromosomes, whereas males have 1 X and 1 Y chromosome, and that determines whether someone is a male or a female. As I mentioned, these chromosomes are in every single cell of our body, and they're packaged very, very tightly into each cell. When you unravel the chromosomes, this is where our genetic information lies. So our genes are composed of DNA, and our DNA and our genes are important because they code for proteins. Genes also code for things like hair color, eye color, but our genes are also important for coding for other things such as just normal growth and development. And so if you have a change in any of these genes, it can affect what that gene is supposed to code for. Going a little bit into more detail, our genes are actually composed of what's called introns and exons. So the exons are the green portion, the introns are the purple portion. The exons are actually the coding parts of the gene, and the introns are kind of the sections in between the exons that don't necessarily code for information, but there still might be some important information within them. But when our genes are being processed, here this is again showing the exons in reddish orange and the introns in blue. When our genes are being processed, the introns get cut out and the exons get pushed together. And this is actually how the DNA or the genes are read. Now, when we think about genetics, I like to use a lot of analogies. The first is when I describe the genome. So you can think of the genome, which is all of our genetic information, our genes, our chromosomes, our DNA. You can think of it like a set of encyclopedia where you have a lot of different volumes. One chromosome is the equivalent to 1 volume or one of those books. A gene, again here with our exons and introns, is like a sentence in that book. And our DNA, or letters of our genetic code, are simply like the letters of the alphabet. So when we think of genetic conditions, you can have differences at any of these levels. So you can have a spelling change, you can have an entire sentence that's missing, you can have an entire volume that's extra or missing, or maybe just a few pages, or you can have more than 1 volume or 1 chromosome involved. This is going to be really important when we start talking about the genetics of HADS and how to specifically test for it, and why some tests might be informative, whereas other tests might not be. Moving on to genetic variants. Genetic variants are simply changes in a gene. We used to use the term mutation, but that term is a little bit outdated, and the preferred term is to say variants. But you still might see the term mutation being used in publications or materials. We all have these genetic variants in our genes. They're what make us unique. Some of the changes that we have, some of these variants may be benign and not really cause any problems. They don't really affect how the gene functions, but some changes might be more serious and can cause problems with what that particular gene is supposed to do. And these are what we call pathogenic variants. Sometimes if we're doing a genetic test and we're reading every single letter of a gene, we find a change in a gene but we haven't seen it before, or it hasn't been reported in the medical literature, and that can make it really hard to interpret. And so these are often called variants of unknown or uncertain significance, or a VUS. We see this very, very often where we find a change, but we don't necessarily know if it's benign, meaning it's probably unlikely to cause any problems, or pathogenic, which means it most likely causes a disease or a condition. So sometimes testing other family members may help us interpret those results. If a parent or an unaffected sibling has the same exact change that the child has, then it's likely that that change is unrelated to what we're seeing in the child, and it's probably a benign change. However, if neither parent has that same change and that change just happened for the first time in the child, then we're more likely to think, could this be the cause of what we're seeing in the child? And then it might be considered pathogenic. When we think of types of variants or genetic changes that we can see, I like to use this example. If we pretend that this is the original sentence or the original gene, our genes are composed of 3-letter words, and they're structured in these 3-letter words. And so that's why every word in this sentence has just 3 letters. And that's going to be important. So if the original sentence is, the cat had red fur and ran far, and then there's a period, some of the changes that we can see, or the genetic variants we can see, you can see a deletion of a letter. So in this case, the D is removed. And when the D is removed, everything shifts over to, again, because it needs to stay in 3-letter words. And so when everything shifts over, then again, you can still see only the D was removed, but all the other letters, sorry, there's a bug, are still here. So red, fur, ran, far. But everything is shifted. So this sentence now becomes unreadable. This is also called a frame shift because you're shifting the reading frame. You can also have a duplication, meaning that one letter is added. And again, if you add a letter, it's also going to shift the reading frame. We can also see missense changes or missense variants. And this is when a letter, for example, this E is here instead of the U. And so that's when there's a single letter change. So one letter gets changed to another. Some changes are not as bad as other changes because you can still read the sentence and generally understand what the message is. Similarly, with genetic changes, some changes might not be as severe because you can still, the gene might understand what it's supposed to do and it might still code for a protein, but it might not be exactly the same. Whereas other missense changes, depending on where it is in the gene, can affect the meaning of that sentence and what the gene is supposed to do. So in this example, the C was changed to an M. So it says the mat had red fur and ran far, which doesn't really make sense. And then a third type of change that we can see are called nonsense changes. And this is when there's a period added into the middle of a sentence where there shouldn't be one. So it's kind of like a stop sign. So this is the cat had red, and then you just end the sentence. These are typically more severe than the missense changes, again, because now you're not even reading the entire gene or sentence. And so these can cause problems as well. Now, as we go to the genetics of HADS, so HADS is caused by variants or changes in a specific gene called EBF3. Now, this gene is located on chromosome 10. This is just a cartoon picture of chromosome 10. We have 2 copies, typically, of chromosome 10, one that we get from our mom and one that we get from our dad. Our chromosomes are separated into a top part, which is called the P arm, and then a bottom part here, which is called the Q arm. P arm just stands for petite because it's the smaller arm. And then Q was named Q because Q comes after P. So the gene for HADS is located at chromosome 10, position Q, 26.3. And this is just showing you where on the chromosome it is. So it's kind of right down here at the bottom of the chromosome. Now, if we were to look at a child with HADS chromosomes, we would not necessarily see this condition because, again, most of the time, the changes in this gene are spelling changes, which I'll show in the next slide. They're going to be changes in the actual gene, a spelling change, rather than a missing part of this chromosome. And so it's like if you were to look at a book, you wouldn't necessarily know there's a typo on page 50, but you would know if there's, 100 pages missing from that book. There are some children and individuals who have a deletion or a missing part of this chromosome area. And so they have EBF3 that's entirely missing, but they also have other genes probably that are missing as well. We would think that these children might have a little bit more medical, developmental, or health problems because, again, they're missing the entire gene as well as probably some other genes that are important for development. However, most of the time, the changes that we see in HADS are spelling changes, not chromosome conditions. This is just showing you where it is. This particular gene codes for a protein that's really important for activating or turning on other genes. And this gene, when we think about where does this gene function in the body, when they've done studies, they've shown that it's expressed at very high levels in the developing nervous system. So the nervous system, the central nervous system, is our brain, our spinal cord. And so that's likely why when we have changes in this gene and it doesn't work correctly, we see that the resulting issues are related to low muscle tone, which is the hypotonia, the ataxia or the unsteady gait, and some delays in development, because this gene is really important in the nervous system, in the brain. And so if it's not working correctly, those are the systems that are going to be impacted the most typically. Now, what are the types of variants that we can see in EBF3? Again, I talked about our original sentence, and so we can see all of these types of changes in HADS. And so we can see deletions or duplications, which lead to a frame shift. Again, everything is shifted over. The most common types of change that we see, though, are these missense changes, where it's just one letter was changed to another letter. But we can also have nonsense changes where there's just a period added or a stop in the middle of the gene. We can also see changes that are called splice site changes, and this goes back to those introns and exons. As I mentioned, when we're processing our genes, we need to take out the introns so that all the exons can go together. Some kids with HADS will have a change right where the exon and intron meet, and so it doesn't know where to kind of splice or cut the introns out, and so that can lead to exons being cut off too short or including more introns than they should, and so that's another type of variant that we can see in HADS as well. But most of the variants, as I mentioned, that cause HADS are misspellings in the gene. And these variants change an amino acid. And so, again, if we think about a protein and a gene, our genes all code for a little amino acids. And each amino acid is like a bead on a necklace. And this final necklace here is the final protein that's made. And so each 3-letter word codes for one of these little amino acids or these little beads. If there is a spelling change in one of these that leads to a difference in that bead, kind of like this, it's going to affect the entire product. And so instead of having a necklace that looks like this, you might get a necklace because of this change here that doesn't know how to fold properly. It might not know how to work as it should, or it might not work correctly. It might only work half the amount of time. And so these are some of the changes that we can see, these misspellings that lead to a protein not working correctly. So what are some of the genetic testing options that we can see for all genetic conditions? And then I'll really focus on HADS. And so again, going back to this analogy of the genome and the encyclopedia, so really the type of tests that you want to do is dependent on what condition you think you're looking for and how that condition is caused. And so if we're looking for a spelling change in the sentence, we don't necessarily need to look at the chromosome at the chromosome level to see if there's an extra book or volume of chromosomes, because that's not what we're looking for. And so I'll go into the different types of genetic tests, just so to give some background. So the first kind of highest level test is called a chromosome analysis. And this involves looking at all of the chromosomes under a microscope. Essentially, you're just counting the chromosomes, making sure there's a correct number and that there aren't any very large missing or extra pieces of chromosome material. One example of a condition that we pick up often on a chromosome analysis is Down syndrome. And so Down syndrome is also called trisomy 21, tri meaning 3 copies or bodies of chromosome 21. So you can see in this picture here where typically we should have 2 copies. People with Down syndrome have 3 copies of chromosome 21. One of the limitations with this, again, though, is that it can only detect relatively large chromosome abnormalities, but will miss smaller ones. Some of your children might have had what's called a chromosome microarray analysis. So a chromosome microarray, or a CMA, looks at a person's DNA and is able to detect very small missing or extra pieces of chromosome material. So instead of entire volumes, we're looking at some pages that might be missing or put in there. So we're able to see much more detail than in a routine chromosome study. But again, we're looking at the chromosomes and to see if there's missing chromosome material rather than at the words on the page. As we get down more into detail, we can do single gene testing, and single gene involves sequencing or reading one gene and looking for the variants or those misspellings in any of the letters. The difficulty with this is that you typically have to know what gene you're interested in, and if you're just focused on that one gene, then you can just send this one test. And this is just showing how, this simple little typo really changes the meaning of the sentence. So instead of saying human brain, it says human Brian is still evolving. These results usually take about 2 to 4 weeks to get back. What we're often doing now, though, in the clinic is multi-gene panel testing. And so oftentimes genetic conditions are caused by more than one gene, or we might think of a condition, but there are probably other conditions that it could be. So we don't really want to just test one gene. We want to look at a panel of genes. For example, a panel of genes looking for seizures, or for kids with developmental delay or intellectual disability, or kids with a certain type of heart condition. And so there are panels available that involve looking at 300, 200, however many genes are on that panel. And so this will identify misspellings in any of those genes that might cause a condition. And this test usually takes a little bit longer, about 4 to 6 weeks. One of the newer tests, probably in the last 10 or so years, that's come up is whole exome sequencing. And so whole exome sequencing examines the coding parts of all of our genes. So again, the exons, which are the coding parts. When we think about our whole genome, the coding parts of our gene, or the exome, only represents about 1% of all of the information. And so there's so much more of the genome that's non-coding. So the exome is really only this 1%, where again, it's only the exons or the coding parts of the gene, not the introns. We have about 20,000 genes. However, we only know the function of about 4,000 of those genes. And so when we're doing whole exome sequencing, we're looking for changes in all 20,000 genes, but we will likely find changes in other genes that we don't necessarily know how to interpret yet. So the interpretation with this can be difficult, especially if we identify variants of unknown or uncertain significance. However, the likelihood of getting an answer or a diagnosis in a child or an individual where we suspect a genetic condition is around 30 to 50%, depending on the patient population and the indication. So for example, if we were to see a child in clinic and we were to order this, the likelihood of us getting a diagnosis is about 30%. Whereas if we are in the NICU and we see a baby that's critically ill with whether they have birth defects or seizures or growth problems, the likelihood that it's going to be positive is probably closer to 50%, given the severity of their condition and the likelihood that it is genetic. And then the last test I want to go into is whole genome sequencing. And so the whole genome examines the coding and the non-coding regions of our entire genome, so the exons and the introns. And that's 3.2 billion base pairs or letters of the alphabet. It also has the ability to detect those chromosome changes as well, excuse me, similar to the chromosome microarray analysis. Clinical availability of this is still limited. However, there are a few labs that do offer whole genome sequencing. And interpretation of these can be really difficult given the uncertainty of the types of results that might be uncovered. And so, for example, if you were to find a change in an intron, typically, again, since these are not coding and they usually get cut out anyway, does this affect maybe this exon, or will it have an impact on the final protein product? This does have great potential, though, as a first-tier test for individuals with suspected conditions, since, again, it will pick up not only these spelling changes, but also chromosome changes as well. And so it's kind of like combining the exome and a microarray at the same time. So genetic testing for HADS, so individuals may be diagnosed by a panel test. So some kids might have had a panel that's specific to developmental delay or intellectual disability or some other neurologic concerns. But you would want to make sure that the EBF3 gene is on that list of genes that is being tested. And so most of the labs will have a list of all the genes that are on their panels. And so making sure that the right gene is on there, or that the gene you're focused on is on there, is really important. But most kids, I would imagine, or individuals would have had whole exome sequencing, maybe whole genome sequencing, but I would think a lot of the diagnoses are being now picked up on whole exome sequencing. Again, since many of these kids might present with some delays in development, they might have had a chromosome microarray, but that would be normal. Again, because there's not typically a missing piece of the chromosome, it's a spelling change within a gene. And so if a child has had a normal microarray, usually in our clinic, our next step, if we don't really have a panel in mind, is to do whole exome sequencing where it might be picked up. Again, the microarray is the least likely to get a diagnosis, since most individuals will have a normal CMA because they don't have a chromosome abnormality. And then the other option would be known familial variant testing. And so this is if you know the variant in your child or in the individual and other people want to be tested, or if you want to test future pregnancies to see if they will have the same change, people can do known familial variant testing, where the lab will look at that one gene and that one specific variant to see if it's present or absent, so that you know if you have the condition or you don't. But this is only used if we know the variant in the family and the family wants to get tested for that condition only. So with all that background, I wanted to just kind of explain what you might see on your genetic test results, since it can be sometimes confusing and labs have different ways of presenting the information, and it's not always straightforward. So this is a sample whole exome sequencing report from a lab. This is not a real person. This is from their website, a sample report. And so I'm going to break this down, but here are a lot of big terms, a lot of terminology. And so I want to break it down with you. This isn't HAD specific, but you might see primary findings, secondary findings, carrier status, PG, discovery findings. You'll see an indication for the test. So this is usually what the ordering provider will tell the lab about why they're ordering the test. This is really important because with a test like exome sequencing, they will be focused on genes related to the indication. So in this particular one, the indication was intellectual disability, mild specific learning disability, delayed speech and language, long face, high narrow palate. So some facial features as well as some intellectual disability. And then they'll usually will have a table showing what they found and some additional information, which I'll get to. And then they'll have a separate section below that with more written information about what they found and what this means. So to kind of break it down from the start, this beginning part. So the primary findings, it says heterozygous for a pathogenic variant in PPP2R5D. So what does that all mean? So heterozygous means that one variant was identified on one copy of the gene. Again, we have two copies of every gene because we get one from mom and one from dad. Heterozygous means that they found one change in one copy of the gene. This is typically what we see in HADS and EVF3. There's one copy of the gene that has the variant and the other copy is normal, unaffected, does not have any changes. Sometimes you might see the term homozygous. Homozygous means that there are two changes, one on each copy of the gene, and they're the same change. So the same exact spelling change on both copies. Sometimes you might see the term compound heterozygous. This again means that both genes are involved, but they're two different spelling changes. So there's two different variants on the same gene, one on each copy. These are more common for recessive conditions such as cystic fibrosis, Tay-Sachs disease, sickle cell disease, where a child is affected if they have two non-working copies of the gene. With a condition like HADS, you only have to have one non-working copy in order to have the condition. So for this condition as well, it's heterozygous. For a pathogenic variant, again, pathogenic means causes a condition or a disease. The other categories that labs will use now, and this is standard nomenclature for all labs, there's 5 different categories ranging from pathogenic to benign. So benign meaning unlikely to cause disease, unrelated, just a normal variant. Pathogenic meaning we know it causes disease. Then you can have likely benign, meaning we don't really know, but we think it's probably normal. Or you can have likely pathogenic, where it hasn't been maybe confirmed, or maybe it's been seen in a couple of reports, but there's not enough evidence to move it to pathogenic. And then right in the middle is where you have variant of uncertain significance, where you don't really know which direction it could go to. So oftentimes the lab might be able to predict if a change in a gene is likely to be benign or pathogenic, if that specific change has been seen before in other children with the condition, or if the specific change has been seen before in healthy, unaffected individuals or their parents. So if it's been seen before in other children with the condition, then it's more likely to be pathogenic. If this change has been seen before in healthy individuals, in unaffected parents and siblings, then it's probably more likely to be benign and not what you're looking for. Now, when we have those variants of uncertain significance, which we see all the time, and they cause us as much anxiety and confusion as they probably do for families, what do we do and how do we interpret those? Well, sometimes what we'll do is test the parents. And so a lot of the time for exome sequencing, we're not just getting a sample on the child, we're also getting a sample on the parents at the same time for this reason. If we were to find a change, we want to see, did this change happen for the first time in the child and it was not passed on from either family member, which is often the case for HADS, or did this change, was it passed on from a parent? So we'll do familial testing to see if that variant segregates or goes along with the condition. The labs also will use different software prediction programs. These are 2 of them that they use that can try to predict how that specific spelling change affects the gene and the protein. They also look at conservation studies. And so this is looking at this particular gene across different species to see, is the location of this variant, is it really important for all these different species? And if so, then it might mean that that region of the gene is really important, and changes to that might affect how that gene works. And then we'll also look at population databases. So this is called EXACT and this is called NOMAD. And these are databases of healthy individuals. And so if you were to see these variants in that population database, you would be probably less likely to think that this is causative of a condition. Going on to kind of the second part of that table of the result is this is really the most important part. So I'm going to break this down. This is showing you the gene. Again, this is the gene name. For HADS, it would say EBF3 here. The mode of inheritance would be, this is AD. That stands for autosomal dominant. Again, for dominant conditions, you just need one copy of the gene change to cause a condition. For the DNA variation, I'll break that down right now. And so, oops, sorry, my animation was off. So again, here is our gene example, or the sentence. And in this example, the cat here, the C, was changed to an M. And so when you look here at this nomenclature, it says C period 598 G to A, which I put here. This means at position number 598 in the DNA code, which is the C DNA, and that's where that C comes from, the nucleotide, or the letter G, was changed to an A. So it's literally just counting the letters. So 1, 2, 3, all the way until 598, where the letter G down here is changed to an A, and that shouldn't be there. So in this example, we would say C for a C was changed to an M. For the C in cat was changed to an M for Matt. So that's what that number refers to. The second part here, the P glue 200 lists. So this means that amino acid number 200 in the protein change, P for protein, the amino acid glutamic acid, which is the glue, was changed to a lysine, which is this list. So the amino acids are the words here. So this 3-letter word is amino acid number 1. This 3-letter word is amino acid number 2, all the way to amino acid number 200. So these are those beads on the necklace. So at this position, where it should have been this amino acid, it was changed to lysine, and that is a different bead, and it's going to change the entire structure of that sentence, that necklace, that protein. I hope that's clear as mud. Some of the reports will also have this highest allele frequency in the nomad population. So what does that mean? So that's, again, looking at that healthy population database to see, has this change been seen in healthy individuals? And this is saying, no, it's not present in that database, or it's not present in the individuals in that database. Again, more likely to mean this is probably a pathogenic or real change. This is just showing those prediction softwares that sometimes they can be helpful, sometimes they give conflicting information, so it's not really helpful in this case. But their interpretation of this change is pathogenic. So if we were to see this in a report, we would think this is our answer. This is the specific genetic change in this child. Why is it important to know the variant? Well, sometimes knowing the genetic variant, or if we're talking about chromosome conditions, the size and location of the chromosome abnormality, can help predict the clinical features that might be more likely to occur in that individual. So when you know the genetic variant, that is called your genotype. And then when you see if it can predict the clinical features, that's called someone's phenotype. And so a lot of times geneticists, researchers, they want to know, are there genotype-phenotype correlations? Meaning, can we predict what the individual features that person might have based on their genetic result? Sometimes we can, and that's why it's important to know the specific variant so that we can see if there are correlations to be made. For example, some changes or variants might lead to a greater risk of seizures, or some variants might lead to a greater risk of heart defects. And so it's important to have that information. Sometimes we can't make those correlations, which is fine too, but that's why we like to know that. There's still a lot of variability though between and within families who carry the exact same genetic change, so we are still cautious about making assumptions. And so even if two children have the exact same genetic change, there's still going to be variability between those kids because, again, they have 19,999 other genes that are also contributing to that person. So lastly, I want to just close on some frequently asked questions that we typically see in our genetics clinic. So what are the chances of having another child with HADS? And so if you have a child with HADS, what are the chances that you could have another child with HADS? So most of the time, the chances of having another child with HADS is very, very low. That means the reason for that is that the individual with HADS usually got that spelling change right at the time of conception as a new change, or what we call de novo. And this was not inherited from either parent. There is nothing at all that the mom did or did not do during the pregnancy to cause this to happen. There's no way that they would have known that this was going to happen. And this change just happened right at conception. So it was not passed on from mom or dad. However, we always say that the risk is very low, less than 1%. We never say zero, because rarely, sometimes the parent might carry that genetic change in a very small percentage of their cells, such as the sperm or their egg cells. This is what we call mosaicism. And mosaic just means kind of a mixture of cells that have the change and some that don't. So if the egg or sperm cell carry the genetic change, then there is a theoretical risk or an increased risk of having a child with HADS, even if that parent is unaffected. Even if we were to test blood and there's no genetic change, they still might have that change in their egg or sperm cell. This is very difficult to detect. We don't routinely test for it, but it's also very rare. But that's why we say the risk is never zero. It's less than 1%, so still very low. Are other family members at increased risk of having a child with HADS? So no. Again, since neither parent has the change, most of the time these occur just by chance in the child, we would say that other family members are not at an increased risk of having a child with HADS unless there is maybe a genetic rearrangement or chromosome rearrangement that led to the condition in the family. And so if a child had their HADS because of a chromosome 10 abnormality, sometimes those can be inherited. And that can run in a family, and it can become unbalanced. But most of the time, no, there is not an increased risk. So the risk of other family members, so your other children, your brothers, sisters, cousins of having the child with HADS is the same as the general population risk or incidence of HADS. So even with that information, you're still wondering, well, what are the reproductive options that are available in future pregnancies? If we decide to have more children, what options are available to us? So I would definitely recommend speaking to a genetic counselor before getting pregnant to discuss the options. This is called preconception counseling. And we have prenatal genetic counselors that are available to do counseling to really talk to couples about what their choices are, not just about for HADS, but also for screening for other genetic conditions, such as Down syndrome, such as carrier screening for things like cystic fibrosis and Tay-Sachs and other common recessive conditions. And so I definitely recommend meeting with a genetic counselor before getting pregnant to discuss those options. A lot of times that's not possible, but that's what we would recommend. The recurrence risk is low for HADS, but some couples still might want to pursue genetic testing. And so there are a few options. So the first would be in vitro fertilization with pre-implantation genetic testing. And this is a mouthful. So essentially this is for couples who do not want to do invasive testing during a pregnancy, but they want to do things before they get pregnant. So the couple will go through IVF, in vitro fertilization, with the sperm and egg, and then they will have embryos that are created from that. And then there are labs that will test the embryos for the genetic condition that is in the family and implant the embryos that are unaffected or do not carry that genetic change. This has been done for a long time now and with great success. For couples that don't want to go that route, there's prenatal diagnosis. And so this is where couples would conceive naturally and get pregnant. And then you could do testing in the first trimester with a procedure called a CVS, where they're taking some cells from the placenta and doing genetic testing on that, looking specifically for the EBF3 change. Or you could do it in the second semester through amniocentesis, which is similar, but you're taking, instead of cells from the placenta, you're extracting some amniotic fluid and testing the cells from that fluid that just kind of brush off the baby's skin. For all of these options, you would need to know the specific genetic variant that you are testing first before doing any kind of genetic testing. With these other options, you can also, the more routine studies are things like chromosome analysis, looking for things like Down syndrome. And so you would need to speak with your genetic counselor to make sure that this test is added on, because it is not a routine test. And again, if you were to do chromosomes would look normal. If you were to do a chromosome microarray, again, that's going to be normal, because this is often a spelling change. And most labs will not just look for this unless you ask them to. And then lastly, I wanted to just share some resources. And so where can I find more information? So the HADS Foundation is a wealth of information, as you know, where they have a lot of information about the condition, resources, support, research, and I think this is a direction where HADS is going with a lot of different research opportunities. Global Genes and NORD are more just general support organizations for rare diseases that have a lot of resources for families that are that have special needs or might have resources and need some support in those areas. And then if you wanted to reach out or find a genetic counselor in your area, you can go to our NSGC, or National Society of Genetic Counselors, and there's a Find a Genetic Counselor link where you can put in your zip code and find a genetic counselor to speak to. But otherwise, you can also just email me. This is my email, my contact information. And I'm so grateful to have had a chance to work at and to participate in the first ever HADS conference a couple of years ago. And it was my great pleasure to get to work with everyone. So I'm happy to take any questions through email. And thank you so much for your time and attention.